Are you ready?
No,
Miss
but we
A.
just start.
Okay. Welcome to Easy Immunology. My name is Stefan.
And my name is Alex, and today we want to talk about how T cells get activated. When we talk about T cells, we need to talk about the adaptive immune system before, because T cells belong to the adaptive immune system, because they are lymphocytes. And we have two different kinds of lymphocytes, the B cells and the T cells. Both are differentiated from a common lymphoid progenitor. When they never saw their antigen before, they are naive, but they can start to differentiate and proliferate into the activated form of B cells or the activated form of T cells. And after their activation, when they are activated by dendritic cells, which we explained in the last video a little bit, but go into detail in this video, they start to do their effector functions. For B cells, it's the production of antibodies, for example, and effector T cells can organize the attack from other immune cells when we have T helper cells, or kill themselves infected immune cells. This is true for cytotoxic T cells.
If we now look at a dendritic cell that comes into contact with a pathogen, for example, this orange bacterium or this green virus, these cells, these dendritic cells, which are called antigen-presenting cells because they present the antigens that they generate from these pathogens, which we look at in the next slide, to the T cells, they become activated. And this is something that we have explained in the last video with all these different pathogen-associated molecular patterns that activate these cells via their pathogen recognition receptors. So if you're interested, we will link the video above. But what we now want to look at is how the antigen-presenting cell activates a naive T cell. So if we have this dendritic cell, this antigen-presenting cell, it can recognize, for example, the presence of a bacterium via structures that only exist in this bacterium. For example, this flagellin molecule that is part of the flagellum, this whip-like structure that allows this bacterium to flow in the medium. And these flagellin molecules are recognized by a receptor called toll-like receptor. And once this flagellin binds to the toll-like receptors, two different things happen. The first is that an activating signal is transmitted into the nucleus of the cell, and this triggers the transcription of many different genes. It can be several thousand different genes that are activated, which totally switches the behavior and the capabilities of the cell to activate other cells. And the second thing that happens is that the cell also takes up the flagellin or even the whole pathogen, and then it ends up in the endosome. This is a compartment in the cell which then fuses with lysosomes. These are other compartments that are full of proteases, so proteins that are specialized in degrading other proteins. And then the flagellin or the whole pathogen, usually it's the whole pathogen, is degraded into small parts of the proteins, which are called peptides. They can be between 8 and 12 amino acids long. And these peptides are then presented on the surface of the antigen-presenting cell in so-called MHC molecules. There it's structures like this, and in the middle we have the peptide, and this is called the antigen presentation. Therefore, the dendritic cell is called a antigen-presenting cells because it takes up stuff from their surrounding, degrades it into peptides, and these peptides are then presented on the surface of the antigen-presenting cell. So by taking up these pathogens and processing them into peptides and showing these peptides on these MHC molecules, the antigen-presenting cell shows on its surface what it has taken up and what it is also dealing with in its inside.
And actually, this is already the first signal to activate T cells, because T cells need 3 different kinds of signals to become activated. The peptide is the first one, and the second one are the co-stimulatory molecules, which are produced after the gene transcription activation which is activated by the toll-like receptors. And then we have also a third signal, which are the cytokines. And the cytokines are showing the T cells in which kind of response and fight the T cell should start against the pathogen. Yeah, and all three signals together activate the T cells and lead to further immune responses.
So Alex explained to you that we need 3 signals to activate the T cells, and this is really important that we have all 3 signals, because the T cells can do a lot of damage in our body, and only if a T cell gets all 3 signals, it is allowed to become activated and to fight the pathogen. And signal 1 ensures that only a T cell that has a receptor that is specifically recognizing the peptides from this blue bacterium get activated, and this ensures that only the specific T cells that we need right now in that moment are activated, and not for example, a T cell that is born to fight, for example, a virus. And with the signal 2, the antigen-presenting cell shows the T cell that the peptides it recognized via signal 1 come from a pathogen that the dendritic cell has decided is foreign and dangerous, because structures from this pathogen have activated these pathogen recognition receptors, like the toll-like receptor, and triggered the expression of these co-stimulatory molecules. So signal 1 and 2 make sure that only specific cells get activated and that they get activated efficiently, but then they don't know what to do because they can differentiate into T cells that make antibacterial responses or antiviral responses, and this information is then transmitted via the cytokines. These are small molecules that tell the T cell via binding to specific receptor on the surface of the T cell that we have here an infection with a blue bacterium, and the bacterium should be fought with a specific response. Therefore, the T cell should differentiate into a specific antibacterial T cell.
Next to antibacterial T cells, we also have T cells which can fight parasites, for example, or viruses.
Yeah.
This picture just showed you in a different way what you just saw in the last slide. So we have antigen-presenting cells, APCs, which get activated by the pathogen-associated molecular patterns from bacteria or viruses, and then activate naive T cells via the 3 signals we just explained to you. And the naive T cell then differentiate into an effector T cell, and these effector T cells then secrete cytokines. And via these cytokines, the other immune cells are activated. For example, phagocytes, for example, macrophages, which start to eat or destroy pathogens in different ways. Eating is one of them. Then we have the activation of neutrophils, which are causing also inflammation. Or we also have the activation of other T and B cells, which then also proliferate and differentiate to effector T and B cells. And altogether, this should reduce the amount of pathogens and is the strategy from our immune system to fight these kinds of pathogens.
With this, we are at the end of our video for today. We hope you learned a little bit about how T cells become activated. In the next video, we would like to look at how B cells are activated. And with this, we wish you all the best, stay healthy, and we see you in the next video. Bye.
Bye.
Welcome to Easy Immunology. My name is Stefan.
No.
It is actually Stefan.
No.
Pretty sure. Let me check my ID.
I know there's 2 more. Okay, again.
Your hair was not correct, huh?
No, it was kitzeling me on my nose.
Ah, okay.
And we have the next outtake.